It is a pleasure to introduce Professor Ioannis Ragoussis, who is the head of the Genome Sciences McGill Genome Center. I just wanted to say a few words about his background and his previous work. He did his master's and PhD degrees in biochemistry at the University in Tübingen in Germany. And since then, he held a variety of appointments at different institutions in England in the first place. He was the head of genomics at the Wellcome Trust Center for Human Genetics at the University of Oxford from 2001 to 2013. Before that, he was a lecturer, senior lecturer, and advanced reader in medical molecular genomics at the King's College in London. And he holds numerous grants, and he is published. He has co-authored or authored more than 200 publications, very well cited as well. And with this, thank you, Professor Ragoussis, for coming to our seminar. And please. Help me welcome. Thank you.
Thank you. It's a pleasure to be here. A little bit on short notice, so I hope you excuse me if it seems a little bit haphazardly prepared, the whole thing, but what I'm trying to do is to give you some background on single-cell genomics in terms of methodologies that are being used. I will give you examples of applications of the technology in medicine in particular. I will touch upon a little bit on sequencing technology, which is also important, and also give you an example of current analytical approaches that my lab is working on. So I will try and zoom through basically what I felt that is relevant to disseminate as a knowledge for this very exciting sort of biomedical but also engineering, in a sense, field. And please interrupt and ask questions during the presentation if something is not clear or you need more information. So just to start, what is the main issue is that our bodies, we all, here in this room, have about 37.2 trillion cells in our bodies. And what is really exciting is that now we are in a position to actually read the genetic information in each one, theoretically, at least speaking, in each one of these cells. But not also the genetic information, but also the derived functional information from all these cells in our body. And as I'm going to demonstrate, this allows now us to look at tissues and whole organisms at a completely different level of resolution and achieve major advances in biomedicine and disease and therapeutic approaches. So I'll give you some examples of that, and also how to develop novel therapeutics, and that will be also part of what I would like to demonstrate. So what's the sort of amazing thing is basically what we can do today is reconstruct the way a whole organism or a particular organ can develop from distinct groups of cells that start changing and developing to give you a whole organ and ultimately a whole organism. So you see here an example where we start with a basic number of cells, and then in development you have branching. Now the transcriptional and functional information of these cells can be disseminated to give you at single cell level of resolution, to give you the whole trajectory of development of cells from one particular group of cells, in this example, into all different types of cells that can be distinguished from each other and would constitute the organism or a particular organ in complex organisms. And as a result, it was chosen by the Journal of Science as the 2018's breakthrough of the year. And since then, this type of technology and all technologies that can give us information at single cell resolution are now incorporated in all major research programs. So you have to do it if you want to publish well, basically. How did it all start? As in many cases, it is basically the solutions that involve engineering and biomedical engineering and biomedicine in general are driven by biomedical, if you want, needs, because that's where a lot of the funding can come from. And one of the major motivations for developing the technologies I'm going to talk about comes basically from looking at the cancer and developing an understanding over the last years, so within the last decade, if you want, that approaches that are not able to increase the resolution at which we can study tumors will never give us answers. And why? What is clear is that when you look at tumors derived from different patients, there is a huge amount of heterogeneity between different tumors, so which we defined as intertumoral heterogeneity. So every cancer is going to be different, and every tumor from one person to the next is going to be different, even if they are classified as the same type of tumor, so breast tumors or ovarian, etc. Now, not only are all these tumors different from each other, going from one individual to the next, but if you look into a particular tumor, you will see that each tumor will consist of different cell types. And it's challenging to analyze and identify what are the different cell types in each of these tumors. And this is defined as clonal heterogeneity. Now, within each individual tumor in each person. To make things more difficult, even if you zoom in and you define what are the clones in each tumor, you will see that the cells that can be defined as a particular clone are also different from each other, and there is basically genetic and non-genetic heterogeneity within the clones themselves. And as a result, it was very clear that tumors needed to be analyzed at single cell resolution in order to understand all these levels of heterogeneity and how they are implicated in disease development, but also how we can target these for new therapeutic approaches. So what was clear is that all our original work was basically relying on taking the material, let's say the tumor, and analyzing things in bulk and try to draw conclusion from looking at either expression of RNA or proteins from bulk material. And the answers were always difficult to interpret or led to misinterpretation. While if we are able to separate all the cells, we can see now what are the characteristics of each individual cell type. How to do that has changed over the years, and I give you some examples here. When I was a student, what I had to do, and I did it actually, I spent a day making my own glass pipettes to pipette single cells and isolate them through micromanipulation. And well, it was fun for the first few days, but then at some point you start losing your patience. Therefore, many different approaches have been developed to allow us to isolate single cells. But what was the idea? The idea was to be able to do it rapidly. So you had to isolate the cells before they die in your hands, first, number one. Second, what was clear is that if you wanted to perform molecular biology reactions in single cells, it had to be done in the lowest possible volume, because otherwise it would be inefficient and expensive. So you can see here engineering solutions that involved, for example, microfluidic approaches, array-based or other, everything that you can imagine, either picking cells individually through machines or collecting them through the use of magnets and magnetic beads that would recognize particular markers. And this approach was very popular and is still, if you want to identify well-characterized cell subpopulations and enrich for them. So that was one thing. And very soon, basically, there were several breakthroughs that were linked to approaches to analyze the DNA and the RNA from these cells. And the first, if you want, published approaches, like shown here, in a Nature Methods paper in 2012, involved the capture of single cells using microfluidic chambers that would allow us to trap individual cells and perform DNA amplification followed by sequencing of these cells. Now, the other thing that needed to happen was to develop and refine approaches that will allow us to look at the transcriptome of the cells. So the molecular biology that had to be applied at macrofluidic scale had to be improved and be very powerful in order to capture from experience about 20 to 30% of the transcriptome. So just to clarify, at the moment, the best technologies will give us 20 or 30% of what is in each cell. How do we get that, though? Is as follows. So we need technologies that will allow us to reverse transcribe, so to make DNA out of RNA, in order to amplify the transcriptomic information, so all the mRNA that is contained within each cell, and sequence it, so analyze it through sequencing. And at the moment, the best way to do it is to convert the RNA into DNA and amplify it, so make many copies out of it, and then characterize this by sequencing. So the most powerful method that was developed involved the following. It came through the use of mouse Maloney virus reverse transcriptase. And this reverse transcriptase has the following characteristics. It uses, we put into the reaction a primer, which is an oligonucleotide that has from 20 to 40 oligo-DTs. So it has a sequence that will anneal to the tail of every mRNA molecule. And every mRNA molecule has a variable length of a poly A tail, so it's just one base. It's based on adenine. And this is used to anneal a primer, so our own oligonucleotide to the tail of mature mRNAs, and then perform this process of copying the molecule, the mRNA molecule, into DNA. And what happens is, if this type of reverse transcriptase is used, it has the property that when it finds the end of the original RNA molecule, it has another activity which is called terminal transferase activity, and it will add nucleotides at the end of the copied RNA molecule. So this is now a DNA molecule, and the enzyme will add bases at the end and extend it. And the whole art was to reverse engineer, if you want, to engineer the properties of this enzyme to add specifically these CTP molecules. So as you see here, we are adding cytosines at the end. And you want at least 3 of them to be incorporated. So this was achieved. And what happens next is, once we have extended this with the CTPs, what is going to happen is we put into the reaction now another synthetic oligonucleotide that has at the 3' end Gs, so which pair with the Cs that we have the cytosine. So it has guanosines, and we pair this with the cytosines that we have incorporated into the end of the molecule. So it forms a double-stranded DNA molecule. And the enzyme recognizes now this double-stranded part and jumps over and starts to copy the DNA molecule that it has synthesized. It displaces, degrades basically the RNA, and copies the DNA molecule. So it removes the RNA and makes a copy of the DNA molecule. And what turned out to be the case is that this reaction was really efficient and represents a major breakthrough in single-cell molecular biology, because this type of reaction is the major type of molecular biology that is being used for the transcriptomic analysis of single cells. And what is demonstrated here is that if you look at the whole mRNA molecule from the end to the beginning, what we see here is basically that this approach here in blue gives us a lot of information that goes over the entire length of the original mRNA molecule, much better than other approaches. So that's a key message about the technology that is being used to become efficient. And here is an example of microfluidic approaches. So this is from one machine I have in my lab for this purpose from Fluidigm, and just gives you the scale that is necessary. So basically what we want to do is to trap single cells in one chamber, you can see here, and the volume in which we trap the single cells is about 9 nanoliters. And then what we want to do is to perform the reverse transcription step, so the step I described earlier, whereby we copy RNA and make DNA out of it, also in 9 nanoliters. So the cells move into a very small chamber, whereby the cell is lysed, and we perform this reverse transcription step in a very small volume. And the next thing was to move basically the material that has been sort of the lysed material into a bigger chamber now, in which we perform a polymerase chain reaction to increase the number of copies of the cDNA we have generated. So of the copied, if you want, transcripts that are now in form of DNA, and we multiply their copies in a separate reaction, which took about 135 nanoliters. So what was demonstrated was that if you were to perform these reactions at the level of a nanoliter scale, that these reactions were very efficient and were able to give you similar information as if you would use 10,000 or 100,000 cells and perform a reaction at microliter level. So we reduce basically the volume by 1,000 to go into nanoliter scale and be very efficient. And further improvements allowed us to use even less, so just starting from 3.6 nanoliters and performing the whole reaction into even 15 nanoliters, as you can see here. So the winning combination, if you want, of these technologies was to combine this reverse transcription reaction. It was called ingeniously SmartSeq and SmartSeq 2 now, and SmartSeq 2.A whatever, but it's called SmartSeq. It's quite smart as a reaction, but what is important is how do you read this information? So we make the cDNA in a very sophisticated way. How can we prepare basically material that can be directly sequenced using new and highly developed and cost-efficient sequencing technologies? And one way, or if you want the first way, that was established in order to achieve this aim, developed also an ingenious process that is becoming increasingly popular now for many applications, which is called tagmentation. And what tagmentation is the following. It uses an enzyme that is called a transposase. The transposase is an enzyme that can be combined with specific DNA sequences, and you actually perform a reaction by complexing your enzyme with specific sequences. And what we do is we put in sequences that can be recognized, number one, and second, sequences that will help us to perform a high throughput sequencing reaction directly from the material generated. So you will see what I mean. So what the transposase does is to actually interact and complex itself with the double-stranded DNA molecule we have generated in the reverse transcription reaction. And it cuts the DNA and inserts known sequences now at the end of each molecule. So we don't need to do anything else. The enzyme does the job to insert these sequences. And what we do is to use now our own primers that recognize the sequences inserted by the tagmentase enzyme to amplify further the material in a polymerase chain reaction and have it ready to be sequenced with our high throughput sequences. So this is a popular approach to perform reactions in a very low volume and be able to sequence the material. How does this look like if you perform this reaction? I give you some examples. So if you do this from single cell, so this is the output of a high throughput Illumina sequencer. We don't need to go into the details, but what I want to say, all these rows represent data from single cells, and what we can see is that actually we can take the information and align it against the human genome reference sequence and what is important is that over 90% of the information we are generating is able to find the corresponding sequence in the human genome reference, and we can identify at least 6,000 different genes in each cell. And what I promised to you was that we should be able to capture, in a sense, 20 to 30% of the information, and it does correspond to this. If we do experiments in bulk, we can find about 20,000 genes in these cells, and indeed we get 6,000 at least of them at single cell level, captured with the methodology I described. So this is very encouraging, and if we project the information that we obtain through this technology to the human genome reference, and I just want to explain very briefly how the reference information looks like. So if you can imagine, this is our genome sequence from left to right within a particular chromosome, and what you can see here in black is basically what we know about the information in our genome. So these are pieces of our genome that are used to make RNA. So the black parts are the parts that are transcribed from DNA to RNA, and are the parts that are then translated from RNA into proteins. So these are very important parts of our genome because these are the ones that produce the proteins that make our body. And what we see here is when we project our data from the single cells, is that you can see, so in brown is the information we obtain through our sequencing. And what you see is basically that we obtain the entire coding information. So all the protein coding bits of our mRNA are actually captured and recreated through the sequencing reactions I have described earlier. And you can see that we can do this in every cell. So the rows represent information from individual cells, and indeed we can capture it. And these are two genes that I have just randomly selected out of the human genome, and I'm just demonstrating that if you look at the information derived from single cells, it is indistinguishable in terms of content from information that is derived out of 100 cells or 1,000 cells. So this would be, if you do it in bulk, you get exactly the same quality of information as from single cells. This is very important because it gives us confidence that we can create very good quality data through this, and we are not actually missing any information. So what is there in terms of which part of the genome is being used to make proteins at single cell level, we can capture it to the full extent. So this is very important, a very important step. The next, after we have demonstrated that this is possible using nanoliter level reactions, the next step was to be able to increase the number of these reactions. So we knew that if we perform reactions in nanoliters, then how do we increase the number of these reactions that can be done in parallel, and therefore characterize a lot more cells, so from a few hundred that I have given you examples for, to thousands or even millions at some point. And one approach was the idea that what you need to do is basically to find a way to encapsulate in an emulsion individual cells with, in this case, beads that will have all the reagents, you need for the reverse transcription. Make an emulsion, as you can see here. So you have oil coming through, and your cells and the reaction coming in another channel, and use a microfluidic device that will allow you to mix the cells and your reagents make droplets and perform key parts of the reaction I described earlier within droplets that again will be just nanoliters. So that's one thing that we need to do. And I'm describing here, I'm not going to go to all the details, but what I want to show here very quickly is that the other thing that needed to happen in order to obtain a lot of information in a very cost-effective way out of thousands of cells now, we needed to reduce basically the number of sequencing, so the cost per single cell, while we are retaining this information. And the only thing I want to demonstrate here is that the main invention, and I'm going to show you examples later, is that now, if you remember, we are using at the beginning of the reverse transcription reaction a primer, so our own sequence that has Ts and will recognize the mRNA tails. But with this primer, we include a sequence that is unique to each one of these molecules. So we put in a mixture of primers that has this part in common. This part with the sequence of oligo-DTs will identify all our mRNAs, but each molecule is going to be different from the other. It will have a barcode that we can deconvolute through sequencing. And if you can imagine, by deconvoluting this information, we should be able to count how many different molecules we are capturing. Therefore, what we do now, and shown here, but I'm not going to go into details now, is that we try to reduce the information and capture enough information from each mRNA in the cell, enough to tell us which mRNA it is, and combine this information from the mRNA with this information from our unique molecular identifiers, which will allow us to count how many copies of each mRNA we have. So I'll give you examples of that. So just if we hold on to that for a minute. And then the other thing that happened was the number of technologies that were developed to efficiently encapsulate cells and perform these molecular reactions increased dramatically. I will go into that in more detail, but this involved two major ideas. One idea was to develop further the encapsulation in an emulsion, whereby you perform key steps of the reverse transcription in droplets that are in an emulsion, or alternatively to fabricate chips with little troughs that capture cells. So that's, if you can see it from above, if you want. So here we have a chip here with nanoliter troughs machined into the chip as an array. These troughs capture a single cell at a nanoliter volume, and we perform our reverse transcription reaction, as you can see here. So this is just an image of encapsulating the cells and performing this reaction. So there were two schools. But what I'm going to show later is each of these has advantages and disadvantages. And one major disadvantage of the methods that use this type of capture of cells in chips or in gels or anything you can imagine is that there is a Poisson distribution. Basically, you throw cells in there. Some of your troughs are going to be empty. Some of the troughs will have 2 cells. Some will have 3. Some will have 1. And you need to deconvolute this information. And what we are going to go through later is to show you that the most efficient way of doing it is to have basically an engineering solution that allows a steady flow of your reagent containing particles, if you want, to meet the cells at a very high efficiency. So you want basically to perform the reaction where your reagents here are shown in beads, meet the cells that are coming in this channel, and that every cell that goes through this channel has a very high chance of meeting our reagents, so that we don't lose cells. So that's, if you can imagine, this is a key point. And the difficulty is that when you have here, as shown here, solid beads, so solid beads, it's not easy to engineer a solution that allows these beads to have a steady flow, because they are heavier than the solution in which they swim, if you can imagine, and the distances between them. So the rate of fire, let's say if you imagine this as a machine gun, for example, is not steady. And the whole trick was to produce reagent bullets, if you can imagine, that are fired from this channel, and we know what the distance, what will be the timing of one bullet versus the next bullet that we are firing to hit each of the cells. And I'll go into that in a minute. So this was a major solution. So when solid beads were used to do this, the efficiency at which the bullets hit the cells was about 10%, which means that you needed to have a lot of cells in the solution to capture a fraction of these cells only. So it follows a binomial distribution, if you want. So what do we use in the lab now to overcome this is a solution that is commercially available through a company called 10x Genomics. And what is the difference is that our bullets are actually gel beads. So they are not solid beads, but they are gel beads that contain all our reagents. So these contain the primers that we need for our reverse transcription. They are in a gel bead. And in this gel, you can see here the droplet, the encapsulation. Here is our cell. Oil is outside, and our gel bead that contains the key reagents is in the middle. And the whole trick was to find conditions under which this gel bead will simply dissolve and allow you to start all the molecular biology reactions that we want and to take place in this solution. And why gel beads are so important? The gel beads are important because once you have gel beads, you can actually find conditions at which they are really swimming with the solution that we have, and you can regulate through the pump flow the rate at which our gel bullets, if you want, hit the cells and encapsulate them. And this is basically it. It's quite simple. You put the reagents versus the cells, and they are encapsulated as I have shown you. And I will give you an example later how this, what are the advantages. I'll come to that in a moment. But the whole trick here is to be able to have conditions whereby the gel beads have a steady flow. And what we achieve in the lab that way is that we can capture about 70 or 80% of the cells that go in are hit by the molecular biology reagent containing gel bead, and where you have a very high capture efficiency. This is important because it means that we can just feed the instrument with a few thousand cells and be confident that we capture most of them. So we don't miss anything. This is very important. And then, as I'm showing here, the molecular biology reactions will happen, but we'll go to that in a moment. So what this includes in terms of molecular biology reaction is, as I showed you before, this is based on the SMART-seq approach with the MMLV reverse transcriptase, with the difference that we include now specific sequences in our key oligonucleotide that is included in every gel bead. So every gel bead has a unique combination of oligonucleotides that have the following information, which is key for us to execute the whole molecular biology reactions and deconvolute the data afterwards. So every gel bead will have primers in the gel that all share a particular sequence, so a code or a barcode if you want, that is common to all our reagents within each gel bead. Okay, so every gel bead will have the same thing, but this sequence is going to be different. Therefore, when we read this information in our sequencing, we can identify all molecules that have been generated within this particular droplet. Does this make sense? So this information is here, and this is coupled with another sequence that I mentioned earlier, which is unique to each individual molecule contained within the bead. Therefore, this is now the information, the sequence that is used to anneal to every mRNA molecule. So this is used to capture mRNA molecules. This information here is different in each of these molecules that capture the mRNA and will allow us to count how many different, which ones and how many different mRNA molecules we have captured with our assay. And this information here is going to be used to deconvolute the information afterwards and identify all the different molecules that we have that have successfully led to a reverse transcription of mRNA and put them together as information that we can assign to a particular cell. So this is a quite complex thing that can be put together, but it allows us to identify where the information, which cell the information is coming from, and how many copies of each of the gene we have captured in this particular scenario. The other thing that's being done to cut the cost is that after we have generated our copied DNA, basically is to trim I don't want to go into all the details, but what we want is to trim the information and come close to this 3' end, but retain enough sequence at the end. So we fragment the cDNA into smaller pieces, and we identify this very variable part at the end of each mRNA molecule that has all the barcoding information, and we just retain enough information here to allow us to assign which transcript it is and where it's coming from our genome. So we trim it, and we'll put here another sequence, which is being used to generate the sequencing information. So how does this look like in the lab? What I'm showing you here is an electrophoresis of all the cDNA molecules that are generated in these droplets. And what you can see here, which is quite amazing, I'm sure the resolution of the screen is not enough, but then you have to believe me, this goes from 500 bases here to 10,000 bases. So basically, if you perform these reactions with single cells, the sum of these reactions correspond to the expectation of all our mRNA molecules in our body. So we get molecules that cover the whole spectrum of what you expect to have. Why is this important? It is important because it demonstrates that the quality of the information we have obtained, so we have been very successful in getting the full information that is contained within each of the cells. And what we do next is basically to trim it down, as you can see here, in this case between 350 and 650 bases. We just trim it down to allow us to sequence a much smaller part of this, and I'll show you how it looks like. Just enough to identify which transcript we have captured, and couple this information with a quantitative, with a barcoding information. So this is how the data look like. So if we take the data, the sequencing data that are obtained using this material, and project it on a whole genome sequence, what you see here is the same gene that I was showing you earlier. And here's the coding information in blue. And we ask, what do we get when we perform this type of reaction? You can see here, this is the gene. It's transcribed in this direction. So here is the end of the mRNA molecule. And you see that what we obtain is information that covers the last part of this mRNA molecule. You see here the last 3 bits of it that are being used to make a protein. So this is our information from this reaction. That's one example. And there's another example, another gene that goes in this direction. Again, we capture the last parts of this information, but it is enough for us to unambiguously identify which gene we are sequencing and couple this information with all the barcoding information I told you earlier to be able to say how many cells have this transcript and how many copies of this transcript are there. Is that clear? So there are 3 main technologies that have been developed to do this. And I'm just highlighting a couple of them. So this is the one that we have just described. It's based on gel beads. And the other approaches are based, this approach is based on solid beads. And this is the difference, basically, is that the capture efficiency, as I told you before, is much less. I will provide these slides to you all, so if you're interested in the details of the molecular biology reaction, you can have a look. But that's the bottom line. The approach that we are using now in the lab is the most efficient approach in order to capture as many cells as possible from a mixture and as many genes as possible per cell. And where are we now? These were the first methodologies that were developed. So this is where we started in the lab with this in 2013. And you can see that we were able to characterize about 100 to 1,000 cells with this technology, and that's where we are now, whereby we are able to characterize between 1,000 and 100,000 cells per sample at an equivalent cost. And further methodologies are being applied to go to 1 million of cells at the same cost. So this is what you can do basically at an equivalent cost, and which is very important for understanding, 37 trillion cells per person. So that's where we are now. There are technologies that allow us to characterize between 10,000 and 100,000 cells per sample. And we are using this technology so you get the idea of the scale. Just give you an example from my lab, where we have now data from about 1,000 different samples and 5 million cells from different tissues. And what is important is this, that I give you here an example whereby we are capturing about 4,000 cells from a sample, and we get about 5,000 genes per sample recognized with this technology. And what is important is here, what I described to you earlier, we have all these barcodes that is in every gel bead. So these are plotted on the x-axis. And what we see is that there are about 10,000 of these barcodes that give us information. And what we are able to do is to plot how many different cell barcodes, so these are the barcodes in each gel bead, are being identified through the sequencing, versus how many RNA-derived barcodes are there in each one of those. So we plot that and we identify, if you want, the juicy reactions that have, in this case, between 1,000 and 100,000 different mRNA molecules captured. And we say every one that has about 10,000 or more is definitely a cell. The rest is debris or background. And we can identify our cells, and that's up to here. And that hits the 4,000 mark. So that's how we do it. We use the information from the cell barcode that is in every gel bead, and then the number of different molecules that carry the same barcode in a cell bead to identify from the sequencing information how many cells we have. So in the next few minutes, I'm going to give you some examples of how this technology is being used. So example number one, the following problem. One of the most treatment-resistant breast cancer is the so-called basal type. So this type of cancer is the worst, if you want, today in terms of the need for new treatment approaches. And what is the issue is that they are all called basal-like. But what I want to show you is that they have different characteristics. These are hormone receptors that are expressed in these cells, but it's a heterogeneous group. So although all of them are treatment resistant, they share different markers on their surface. And we need to find new ways of identifying drugs to stop basically these cancers and cure the breast cancer that we see in these women. And here is a collection of these tumors, and this is from Morag Park's laboratory here at the Goodman Cancer Center. And what they do is to take the tumors from the breast of the women and grow this in mice. And this is a collection of tumors, and they test drugs. And you can see here that although these are all basal type tumors, when they are exposed to a particular compound, some of them are completely resistant, they don't understand anything, and some are completely sensitive to the drug. And the question now is, how can we identify which tumors to apply the therapy to? How do we do that? If we do conventional experiments that are shown here, the question will be, is there a relationship between the drug we are giving and the target of how much target of the drug do we have in our sample? And what you can see here is this is the amount of target, which is a receptor, the EGFR, the epidermal growth factor receptor. And what we see is that basically there is no relationship whatsoever between the levels of the target protein in each of these cells and the efficacy of the drug. Therefore, something else must be a factor in making the cells sensitive or insensitive to the treatment. And we need to find what is the characteristic that will allow us to identify which tumor and predict which tumor is going to be sensitive to the treatment, so that only these women will be treated with this drug. And what you can see here is when we analyze these tumors at single cell level, this is what we see, is that actually there are individual cells that express the target of the protein in blue. So in orange, you can see all the cells. And in blue, you can see that it's only some cells that contain the target for the drug. So once we see this, what we need to do is to identify which cells are these cells, what is the characteristic. So we perform the reactions I explained to you earlier, and what we find is that by using the transcriptomic information, we can characterize these cells in different types of cells that start from stem cells. So these are the cells that could give rise to all the cells in the mixture, to fully differentiated cells here as basal cells. And when we combine this information, we find a so-called trajectory whereby we see that these are the cells that are generating all the cells, and then how the cells change and form two distinct types of cells in our mixture. And when we look at these cells in more detail, we can confirm the data that we generated. The cells that are there can be divided into cells that have high levels of the target and cells that have low levels of the target. And to cut the long story short, what we see is that the cells that have high levels of the target are the ones that can proliferate and give rise to the tumor. So basically, if we take these cells out of the mixture, they can recreate the tumor in mice. And what is important is what we can demonstrate next. Basically, if we take the cells, they give rise to the tumor, and we give the drug, the tumor goes down. And what was demonstrated is that you can do this ad infinitum. So basically, you can control the tumor growth in this type of tumor. So what is the strategy that is being applied at the moment? And this was one of our first publications, is that we have a whole setup of a system whereby the breast tumors are propagated in mice. They are analyzed at single cell level. The cells that matter, in this particular case stem cells, are identified, so they are shown here, using single cell genomic technologies. A drug is then given that will stop the proliferation of these particular cells and control the growth of the tumor, as you can see here. And what this does is that this information is being now used in the clinic to treat the women in this case. So I'll stop here with the technology, and if you have any questions so far, let me know.
Questions? A few minutes. I know it's late, but a few minutes for questions.
So I have two questions. The first one is, so if I probably missed it, but if we pile up those data from single cells, we get something similar to the bulk RNA-seq? And the second one is, do we actually have now any technique that I can sequence a cell, not a bunch of cells with single cell resolution? If I see a single cell under microscope, I want to sequence that cell.
Correct. Very good. So if I go back very quickly. So to your first question, indeed, there are two things that come out of this work. One, that if you put the information that comes from each individual cell, so if we synthesize the individual data from each single cell, and this is one of our controls that the experiment works, we always check that the sum of the single cell data recapitulates the bulk of the corresponding cell. So what you asked is a very important control, and it's the number one thing we need to do each time to actually demonstrate that if we synthesize the data derived from the single cells, we get the expected, if you want, data from bulk of the cells. And indeed, this is what happens. So because we have reached a level where the molecular biology applied is as sophisticated as possible today, we reach this point that you put the single cell data together and you recapitulate the bulk data. What was your other questions?
Sequencing a single cell. So can we sequence a cell if I'm
Yes. So
one cell?
if there are methods where, like the one I described here, in this case and in similar instruments, what you can do is you capture the cells in a chamber of a chip, shown here, and you can actually visualize it. You can interact with, you can label it with dyes. You can label it for particular markers from the cell surface. And there are many approaches to capture single cells in microchip that, like David Younger's group is developing, whereby you can look at the microscope and see actually the cell that you are going to analyze. So originally what we did was to capture this information, visualize the cell, and then decide which of these we are going to process for sequencing. But at the end, it turns out that it's more useful to bypass this in most cases and capture as many cells as possible, which you cannot visualize, because if you go for 10,000 or 100,000 cells, this is difficult. But there are methodologies nowadays that try to do this, to rapidly capture information that can be visualized and to which you can refer your sequencing data to at the end. So you're absolutely right. So this is the most desirable feature. And we hope to have this type of technology that allows a mass-scale visualization of cells and then linking the property, the information about the property of the cell that we can capture to the function information we can obtain from the cells.
Thank you. Right, any other questions? Now, please thank the speaker again. Thank you very much for
Thank you.